Thanks, Datsa. Thanks, Marta. And thanks, Olaf, for organising today.
You can use this if you want.
Okay, thank you. So the BOLD signal that we're all used to in fMRI is often used to infer differences in underlying neural activity as a function of, for example, age. And that might be differences in activation or functional connectivity. But some of the effects, for example, of age could reflect the vascular components of the hemodynamic response function without necessarily reflecting any age differences in the underlying neural activity. So that's the problem. We want to use fMRI or the BOLD signal in particular to localize in the brain and infer something about neural activity and how that might change with age, but it could be that we're seeing differences in vascular effects of age. Because as you know, in this schematic, the signal we get out at the end, the BOLD signal on the right here, is actually a cascade of processes starting with a stimulus or a task that does cause changes in neural activity, but that in turn leads to a release of vasodilatory signals such as nitric oxide, which then increase the blood, the capillaries, and that increases blood flow, blood volume, and blood oxidation level, which is ultimately detected by the MRI scanner. And although we want to make inferences about age on neuroactivity, of course, age could affect one or more of these intermediate stages as well. So we have a cohort here called CAMCAN of about 650 people across the adult lifespan from 18 to 88. And we did lots of things with these people, but one thing we did was a sensory motor task where people saw bilateral checkerboards. At the same time, they heard binaural tones. And when this occurred, they had to press with their right index finger on a key. This is a sensory motor task. This was used in one of the first publications. In fact, this publication was actually done halfway through acquisition, so only half the data at this point, but led by Carmen Setvanov. And so we're already aware that this was an issue. And Carmen described a way of adjusting your data for these vascular confounds. And he focused on a measure called RSFA, or resting state fluctuation amplitudes, and I'll explain what this is in a moment. But think of this as a scaling factor that you apply in every voxel to try and remove vascular effects. So here's a standard statistical parametric map that's been thresholded for age effects. So in blue are areas that are more active in young than old, and in red are areas more active in old than young in a sensory motor task. So obviously you have visual cortex, auditory cortex, and here, showing effect of age, is ipsilateral motor cortex. And this is a standard analysis, and from this you might conclude that older people have less neural activity in their visual cortex when they look at these checkerboards than do young people. That's kind of standard analysis, but if you do adjust your data by this thing called RSFA, which I'll explain in a moment, what you'll notice is a lot of these activations disappear, a lot of these age effects disappear. So in visual cortex, for example, if you hone in here, the blue is a standard analysis where you just look at the overall BOLD signal, that declines with age. If you adjust for RSFA, the red line, the age effect is no longer significant and dramatically reduced anyway. It doesn't abolish everything. So in the ipsilateral motor cortex, which actually shows increased activation with age, that is true both without scaling in blue and with scaling in red. So scaling doesn't remove everything. Now, what is this scaling factor? The RSFA is actually nothing more than the standard deviation of band-passed BOLD data from an independent resting state scan. It's supposed to measure vascular activity. But if you're going to adjust your task data by this independent resting state data, you've got to be very confident that your resting state data doesn't include neural differences as a consequence of aging. That's only measuring vascular differences. Otherwise, you'll be throwing the baby out with the bathwater if you try and adjust by RSFA. So Carmen's main job in his paper was to ask, is this RSFA scaling the right thing to do? And for that, he used a mediation analysis. So he took the RSFA from resting state. So this is a voxel-wise measure of the standard deviation of the time series after bandpass filtering. And we know that age affects that. And in CAMCAM, we have a large number of vascular measures. We have blood pressure, we have ECG, and you can do a PCA or extract a number of components that indicate vascular health. And these vascular components do significantly mediate the effect of age on RSFA. So RSFA does capture some vascular components. But the crucial question was, does it also capture some neural components? Because if it does, then we shouldn't use it to scale the data. Unfortunately, in CAM-CAM, we also have MEG data, which is a more direct measure of neural activity. And we took lots of different measures of MEG, power spectra, different sensors, channels, etc. But in no case did the MEG measures of neural activity mediate the effect of age on RSFA. So it appears that RSFA is a relatively pure measure, or at least the age component of RSFA is a relatively pure measure of vascular rather than neural activity. So you can use it to correct your data. So to summarize, many age-related fMRI activations or deactivations are likely to be vascular rather than neural, because when we adjust for RSFA, a lot of the effects disappear. And so one approach to this problem is to adjust your data by RSFA. You might think that functional connectivity, for example, the Pearson correlation coefficient, which is often used to measure functional connectivity, would be independent of these vascular scaling effects, because Pearson correlation is independent of scaling. But actually, in other studies, which I won't show today, we found that functional connectivity is also affected by vascular health. So any effects you find in functional connectivity could be vascular effects rather than neural effects. And that was shown in this paper here by Linda Gierlix, if you're interested. But what I want to do today is another approach. Rather than trying to adjust your data, so either scaling by RSFA for activations, or in the case of functional connectivity, we recommended adjusting by mean connectivity. Rather than doing that, you can have a more complex model of the data, particularly a biophysical model of how the BOLD signal is produced. So rather than change the data, you have a more complex model. And this is a preprint currently under review. And I showed that schematic of the cascade of events between neural activity and BOLD. This is the same thing now, but in terms of differential equations that relate ultimately neural activity, which we're going to measure at some point in time, to the BOLD signal we measure with fMRI. So again, that leads to neural activity leads to a vasodilatory signal, which controls blood flow, that changes blood volume and decreases hemoglobin, and ultimately modulates the BOLD response. And there are a number of parameters in this model, and some of these are quite well known, since they have quite tight priors from animal studies where you can record these parameters in the rodent, for example. But other ones we argued might be affected by aging. And particularly, we focused on three parameters: the transit time of red blood cells through the capillary bed, the decay rate of this vasodilatory signal, and the neural efficacy, which is what we normally care about. We normally want to estimate this, but we're allowing age to affect these three parameters here. And this model has this particular form, so it's just a large number of differential equations that relate these parameters. And this has been quite well studied, as I said, in the animals and in humans. It's also, by the way, the human model used for SPM's DCM, which we heard about earlier, or at least, no, we didn't, for fMRI. DCM for fMRI uses this. So now we've got the full data set of approximately 600 people with usable data, same task as before. This is a maximal intensity projection of the activations in this task. So you've got visual cortex, you've got bilateral auditory cortex, bilateral motor cortex, and SMA, which we're not going to consider here. These are all activated in the task. And I'm going to focus on 4 ROIs: visual, left auditory, left or contralateral motor cortex, and ipsilateral motor cortex. And if you average over all participants, so average across age, and look at, for example, the left auditory cortex, this area here, you see a very canonical hemodynamic response function. So this is using what's called a finite impulse response basis set. So sort of 1 second time bends. You can see it peaks at about 4 to 5 seconds after stimulation, with an undershoot that can last up to about 25 seconds. So that's the average BOLD response to this impulsive stimulation. These are very brief stimuli. In our model, so this is our generally a model where we're fitting each time bin on average across people. We can also look at the effect of age, or the linear effect of age, on each time bin as well. And if you plot that from the same voxel, now looking at the effect of age, the scale has been zoomed up a bit, but you might be able to see this looks like a temporal derivative. So if you imagine adding this bottom plot to the top plot, the result will be shifted later in time. So as you get older, the hemodynamic response becomes more dispersed or sluggish, if you like, over time. So here's the 4 ROIs, or the 4 color maps. We have age going down from young to old on the y-axis, and on the x-axis is peristomus time. So this is a heat map. So you can see as you get older, a tendency for not only a weaker, but also a more dispersed hemat response function. And certainly in the 3 ROIs here, the right motor cortex is quite interesting. It shows a different pattern with a slight deactivation in young people that becomes more of an activation in older people. We'll come back to that. These are the same data, now plotted in tertiles. So the lightest color is young, and then middle age, and then dark color is older people for the 4 ROIs. The dotted lines are the finite impulse response fit, so they are the same as in the color maps above, just binned into age groups. And the smooth line is the fit of this hemodynamic model that I mentioned earlier. And you can see it's not a perfect fit, but it does capture the basic components of the hemodynamic response function, including this strange deactivation in young people in the right motor cortex. Well, not strange, but characteristic deactivation of the ipsilateral motor cortex during motor actions. So the model fit is pretty good. But what we really care about is which of the parameters of the model, I mentioned there's 3 parameters, which of these parameters need to be changed in order to fit the data. So now the 4 regions again, and here we have the age effect as a Z-score, and this is after using something called parametric empirical Bayes. So this is basically using the group average as a prior on the individual participants. And this PEB framework, you can basically shrink parameters that aren't necessary, or get rid of parameters that are not necessary to improve the fit. And what you can see is, on the x-axis are the 3 parameters: neural efficacy, which is what we care about, the decay of the vasodilatory factor, and the transit time. And the first thing to note is that in 3 of the ROIs, there's no need in the model for age to affect neural activity, except in the right motor cortex, where there is a need to the model to increase the neural efficacy parameter as you get older. But to fit those dramatic changes in the HRF shape, instead it tends to increase the rate of decay of the vasodilatory signal. So the decay parameter is positive in all ROIs. And it reduces the rate of transit time. Reduces the transit rate. In other words, makes the transmission slower through the capillary bed with age, at least in 3 of the ROIs. So consistent effect on the transit rate, the decay rate, and an effect at least 3 of the ROIs on a slower transit time through the capillary bed. So in other words, these are vascular components that age is affecting, not neural components. And this resembles the approach that I showed you earlier, where we adjust the data. So if you scale by RSFA, you'll notice that these age effects in visual cortex have largely disappeared, whereas the age effect in the ipsilateral motor cortex, the right motor cortex, hasn't changed after scaling. So the modeling results are consistent with the previous adjustment of data that we did. So to conclude, using biophysical modeling or hemodynamic modeling suggests that many, although not all, age-related fMRI effects reflect vascular rather than neural differences. So while age did affect the hemodynamic response shape in all ROIs, it didn't need to, according to this model, the neural efficacy parameter didn't need to change in 3 of those ROIs. It only needed to increase in the ipsilateral motor cortex, because that is a true neural difference rather than a vascular difference, or at least as well as vascular differences. Instead, age increased the rate of vasodilatory signal decay and decreased the rate of transmission through the capillaries. Now, at this point, I'm not going to try and claim that age has no effect on neural activity in sensory cortices like visual cortex or auditory cortex. The model is saying that it doesn't need to change that parameter. But we actually have MEG data on the same people. In a previous paper by Price et al., we did find effects of age on the latency of evoked responses recorded with MEG. So I'm not claiming that age has no effect on neural activity. What I'm claiming is that those changes in latency are so small, at the order of 50 milliseconds, that they're just not detectable with fMRI. Because fMRI is integrating over seconds, in fact, of neural activity. And the argument here would be that that part of neural activity that's detectable with fMRI may not show strong effects of age. So that's the summary is that if you're doing aging research with fMRI, you need to consider vascular effects if you want to interpret things in neural terms. You can adjust your data in the first paper by an independent measure from resting state data. Or if you're interested in connectivity, you can adjust for mean connectivity, which the Gillick's paper, which I didn't go through, argues can remove vascular effects on connectivity. Or you can take a more, at least for activations, you can take a more modeling-based approach and actually separate out neural and vascular parameters. So that's the take-home message. Or, of course, use a non-hemodynamic measure like MEG. So that's it. Thank you very much.
Thank you, Rik. Any questions for Rik?
Nice, Rik. So when I look at patient population, I of course interested in both components. But if someone only interested in healthy cognitive neuroscience, do they have to do both functional MRI and resting state in order to answer the question correctly?
Or they use a biophysical model. So you don't have to use the resting state. If you're doing a modeling approach, of course you have to trust your model, and there are some issues about which parameters you allow to vary with age. So if you want to look at the preprint, the supplement material is massive because we tried lots of different parameterizations of the HDM. But if you accept the assumption of the model, then you don't need to collect independent data. You can just apply the model for your, certainly for task evoked responses. For connectivity, yes, if you're going to use something like DCM, which has this model in it, your connectivity estimates will also be less biased by vascular components. In fact, Carmen has another paper, which I didn't mention today. He tried to relate functional connectivity to cognitive scores. And if you did standard functional connectivity, like Pearson's correlation coefficient, he found only a very weak relationship between connectivity and cognition. When he applied DCM, which allows for these age-related vascular changes in human response, he got much stronger relationships with the underlying connectivity parameters now and cognition. It's just that removing these vascular source of noise improves connectivity as well. So yeah, I would, if you really want, if you think there might be vascular differences in your two populations, and you haven't got resting state data, then yeah, maybe consider modeling.
Sorry if this is a naive question, but if you just include age in your simple model when you're analysing the results
Yeah.
at the end,
Yeah.
how much of the work does that do?
Well, that will potentially remove vascular and neural components, so you might get nothing left. So
Sure.
so yeah, I mean, but actually, in this case, we are actually interested in the effect of aging. So
Well, that's what I was
if
gonna
you're
say,
interested in
because
how,
you weren't interested in the effect of ages, but you were worried about an age difference. Is what
oh, I see what you mean. So if you're listening like task activations have it, and happen to have a different in age groups, you want to get rid of that. Yes, that's a different question. Sorry, I should have prefaced this talk by saying, if you're interested in studying aging,
Yeah.
if age is a compound no interest, yes, you can adjust for that.
Any other questions? Well, then thank you.
Okay.
And it's-